in general?
Well, I was always interested in science because for some reason I was always curious about how things worked and what is behind their mechanism. For example, here at the Kids Museum, you get to see how electricity works, how the circuits are wired, and see how, for example, that wind works. So I was always curious, and I always asked why, how, and that was really the beginning to my research project. So I was interested in color vision, so I asked, how does this work? And thankfully, I was able to find an internship here.
Okay, so is there a special person or something that happened that inspires you to study neuroscience and color vision?
I actually have a friend and teacher who are colorblind, and from that experience, I was always curious about how they'd see the world, and since as we see the world with so much color, I always wanted to let them see the color too. So I applied for an internship at the National Institutes of Health to specifically learn about color vision, and that's what really brought me through the door. Well, I would first have to thank Dr. Whaley for giving me this opportunity to have an internship at the National Institutes of Health. For my first year, actually, I was an unofficial observer in the lab, and it kind of got me more interested in what they did. So in the second year, which was last year, I applied for an official internship, and I was able to do more research about the RI and color vision. And thanks again, Dr. Whaley.
Okay, so how was the experience in?
It's very different. So in the classroom, you get to learn about the basics. So you probably would learn, for example, an analogy would be, you learn the alphabet. But to apply the alphabet to words, you would have to read. So it's kind of like research. In the classroom, you'd get to learn about the cell, you get to learn about the basics of biology, but you wouldn't really be able to apply that knowledge until you have a hands-on experience, like in the lab or here in the Kids Museum.
Okay, so that was really, my next question is also related. So what was the most important thing you learned from real lab experience at internship?
So personally at this internship, I learned about how we're able to perceive the color blue. So I specifically looked at the cells and how they communicated with each other. And this is a really important foundation for future color vision treatment or color blindness treatment. And I also learned how important it is to persevere. Since looking for the blue specific cells, they're very rare, so I spent a lot of my time just looking for them. And it was kind of like looking for a needle in the haystack, except I didn't know the shape of the needle. So I spent a lot of time persevering, or looking for it. So I really realized how important to persevere no matter how big the obstacles are. And that also kind of lets me realize how important actual research is. Like in the classroom, you probably learn about these famous scientists like Watson Crick, and you probably only understand their basic contribution. You might not actually realize how much effort or how much time they put into it in order to get that final result.
Must have been hard. So what was the most challenging part of your research project, and how did you overcome the challenge?
So the most challenging part of my research was first understanding the images that I had. So I had a large data stack, a special type of electron microscope, and I would first have to understand how these images work. And the second hardest part would be finding the blue specific cells, since they're extremely rare and we don't know much about them. So I spent most of my time looking for them and finding a difference in how they vary from other regular cells.
Can you tell us more about your research project?
So for my research project, I looked at the specific retina, or a layer of our eye, and this area is important for our ability to see different colors. And I specifically looked at the cells that allow us to see the color blue. As we can see, there's a lot of things that are blue. So we don't really know how these cells communicate with each other, and that's what I really looked at. Processor. This creates a really important foundation for color blindness treatment, or our basic foundation of the knowledge of color vision. So this could have many applications.
Nice to know that. And what's your personal plan for the future?
My personal plans would be definitely pursuing research in the future and also learning more about neurobiology, neuroscience in college.
We know that research and studying science take a lot of time. How do you manage your time, and are you participating in other activities such as music or sports in the school or out of school?
Well, I have a lot of extracurricular activities outside of research, so for the majority of my project, I did it over the summer, but I'm still working on it currently throughout the school year. And my other interests would be playing the flute and piccolo. I have a variety of bands that I go to, and I also sometimes act as a bilingual master of ceremonies at local Chinese festivals. And I am also an editor-in-chief at a local Chinese newspaper.
Interests. So do you have any advice for others, especially the younger kids here who are also interested in science?
Definitely. If you like something, pursue it with your whole heart. And when you have obstacles that you face, don't be deterred by them. Continue, and one day you will find the final fruit of your efforts.
Thank you. And I also have a few questions for Dr. Lee. So first is studying neuroscience, and how did you meet Yizhen as an NIH intern?
Well, thanks. First of all, thank Claire for the invitation to be here, and this is my first time visit the museum, and it's a wonderful place. I think it's the place really can trigger curiosity from the kids, and personally, my scientific career started being curious about how we see and how our eye works. Because my mom is an ophthalmologist, and so from very young I'm always curious. I was always curious how people see things, so I always wanted working on vision. And when I had the chance to look at the neural tissue in the back of the eye, the retina, the beauty of the tissue just struck me as something I want to work on. Very much like everyone else, we have to bring the kids to school and go to work. And it just, the work is different for different people. And for us, I think we're lucky that we have the opportunity to sort of explore the unknown as our routine job. And it's quite fascinating, particularly to me also. The other part is having a student come along, and a student like Yizhen that really pick up the project and run for it, and I think that the next generation of scientists, that's another part of being in an institute, not just doing the research but also mentoring the next generation of scientists. I think I see the museum is doing the same, and these are young kids that are very curious about things, how things work, and these are the most important characteristics for being a successful scientist.
So, what do you like best or worst about being?
I guess the best and worst is pretty much the same because doing science, you're exploring the unknown, that the success rate is pretty low. Because of that, sometimes it could be frustrating. But on the other hand, and because of exactly that, when you have success, that's very rewarding and joyful. So I think it's the same thing.
Very interesting answers. Then, how much time do you devote to your research?
I spend quite a bit of time, not only this time that we are working in the lab together with students and postdocs, but you also do a lot of thinking while you're doing other things. Sometimes it's ironic that most of the important discoveries coming from the time that you're maybe just doing your housework, but you have a mind on the subject that you're working on, that serendipitously you have the thoughts and might be most important. our lab mostly working on vision and how particularly the retina, the neural tissue in the back of the eye encode visual information. But I'm interested in neuroscience in general, so how the brain works is the most fascinating idea that mankind has, I guess, to figure out how our own brain works. To me, that I just use retina as a model for our brain because it's approachable and it's has its feature that's suitable for study the brain. So I guess the long-term goal or the wish will be that one day we can figure out how our brain works. Still a long way to go. Quite different because normally PhD student and postdocs, they're fairly well trained and has a pretty good solid knowledge of the subject that we're working on. And for some intern like Yizhen from high school, we really need to brought them up to the level that they could understand the question we're looking at. But good thing is kids that apply to the NIH some intern program, they are all from very good schools and they're very bright kids. And it takes a little bit of time, but they can eventually get the idea. And for instance, Yizhen really quite independently pursue the subject and get the result way beyond our expectations. So it challenged us to explain things that's fairly complicated in a more easily understandable way. I think it's the same as all the volunteers here. You guys are doing and trying to explain things to young kids, how the circuitry works, how the power, electricity, sounds works. I think the same challenge, but it's really worth doing.
I think Yizhen said quite well earlier. One, you have to be very curious and you really want to know. That's really the driving force for any discovery. And second, you have to be very forceful and really persistent on what you are, to the question you wanted to answer. It really sometimes depends on how much effort you make and how deeper you want to dig, and that makes a difference between different researchers. But out of everything, curiosity is the most important.
3D models of these cells in order to actually see how these cells interact directly. And at the beginning in the classroom, I would only learn about the cell. I'd see colorful pictures of these cells, but I wasn't able to actually truly see how these cells looked. So if I can get this to work. One of the cells that I reconstructed. And the first time I reconstructed and made one of these cells a 3D model, I was really amazed by how thousands, millions, or billions of these cells create us. And these tiny structures are able to have such a large impact on us. This is another cell that I reconstructed. And these little cells, this is just one of them, and in that same area, there's hundreds more. And this is like a dense mass of cells. What I actually looked at was just this one small area here, these blue specific cells. And in order to find these cells, I spent a really long time in order to find a difference between these cells and the rest of the cells. And as you can see here, there's a large dense mass, and this is where these cells communicate with each other. So these are the blue specific cells. And in this area here, there's multiple cells and they communicate with each other, and that really struck me as really interesting and cool because these tiny little structures are able to communicate with each other, send signals, and allow us to function properly. Yep. And thanks again for having me here. And thanks, Dr. Whaley, for giving me this opportunity.